Okay, so it is going to be only because of your ptyalin or your salivary amylase. Okay. Now the second thing that is going to happen, guys, once the food has been chewed, okay, it is going to be masticated and then it is mixed with your saliva, and now it is going to be converted into a small mass, okay, and that small mass is called as bolus. Then after that, the once we have this bolus, it is now going to be swallowed down. The process of swallowing is called as deglutition. Okay, fine. After deglutition, the food is now going to enter into your esophagus. See, at this time of, at the time of swallowing, what did I tell you guys? See, the epiglottis is going to be closing off the opening of trachea. And so the food is not going to enter into your trachea. It can enter only and only in the esophagus. Okay. So we said, okay, chalo. So once the food is there in the esophagus, is esophagus really going to help in digestion process? No. Okay, what is it going to do? It is going to only help in the conduction of food from the mouth into the stomach. That's it. Okay, and how is it going to do that? It is going to do that with the help of peristalsis, meaning with the help of this waves of alternate contraction and relaxation. Okay, fine. Chalo. But now after that, we said, okay, we are going to go into the stomach. In the stomach, what are we going to encounter, guys? We are going to encounter our gastric juice. Okay, now gastric juice, what is this going to be made up of? So we have the components. We have huge amounts of water, we have mucus, we have salt, dilute HCl, and then we have two of those enzymes. Okay, see, pepsinogen and prorenin, these are inactive ones, and these are converted into the active ones in the presence of dilute HCl. So pepsinogen is converted into pepsin, prorenin is converted into renin. Okay, then we said, what are the functions of your dilute HCl? What is it really going to do? Okay, so we said, okay, so this dilute HCl, first thing, it is going to stop the action of ptyalin. Okay, so ptyalin was the one which was found in your saliva. So it is going to stop the activity of that. Second thing, whatever germs that are entering through your food, they are going to be killed because of the high acidity of HCl. Third, we are going to see that it is going to soften the food, and while softening the food, it is going to help in breaking down the food. Okay, and the next two things we are going to see is it is going to activate. Okay, activate those inactive enzymes. Okay, so let's see what is going to happen. So we have this prorenin. Prorenin is converted into renin. Okay, but what is this renin going to work? Renin is going to work on your milk proteins. Okay, what are milk proteins? Milk proteins are called as casein. These milk proteins, these are actually soluble in milk. You cannot see it. Okay, so what we need to do is we need to remove them out from the milk, and then only we can digest it. It is that way. So what is renin going to do? Renin is going to convert the milk proteins into your paracasein. So casein to paracasein. And what is this thing? Soluble into insoluble. Now, once we have got this paracasein, which is insoluble, this is going to be in the solid form. This can further be acted upon by your pepsin. Okay, but what is that reaction? Let's try to see. So see, we are going to have pepsinogen. The pepsinogen is, someone is unmute. Okay, so we are going to see, guys, that this pepsinogen is converted into your pepsin. But pepsin, what is this going to work upon? Pepsin is going to work upon your proteins. And when it works upon proteins, what is it do? It's going to control your proteases and peptones, right? So see, pepsin is going to break down these proteins and convert it into smaller proteins called as proteases and peptones. This is what we had seen in the last class, guys. Okay. Sure. Now, the next thing, guys, we will quickly move ahead and we'll see. So, guys, when we talk about the food, how long is the food going to stay in the stomach? Always remember the food, it stays in the stomach for around 3 to 4 hours. Okay. Okay. It stays in the stomach for roughly 3 to 4 hours. And guys, during these 3 to 4 hours, what is going to happen? This food is going to be converted into a paste-like material. Okay, and what is this paste-like material called? Guys, this is known as chyme. And always remember, at this moment, this chyme is going to be acidic. Okay, later on, as we move ahead, we are going to see a few things that are going to happen. Okay, so this is what is going to happen with regards to your entire stomach. Okay, so guys, let me just pull out this picture once again. So at this moment, guys, see, we are out over here. Okay, so in the stomach. Let's try to move down and let's try to see your duodenum, guys. And let's try to find out what is present in the duodenum. Okay, and how it is going to be. So after your stomach, guys, we are into your duodenum. Okay, so let's try to see what is going to happen. Okay, so guys, see, we are somewhere out over here in this region. Now, if you remember, guys, in this region, what have we said? This duodenum, what is this going to receive, guys? It is going to receive the pancreatic duct and it also receives the common bile duct. So you see two things entering out over there. Okay, sure. So let's try to see this duodenum. I can say it receives two things. Okay, it receives two things. One, we have the bile duct, okay, the common bile duct, and the second duct is your pancreatic duct. Okay, but now, guys, let's try to see. We are talking about the common bile duct first. Okay, let's try to talk about this. Then we'll talk about your pancreatic duct. What is going to happen with regards to that? Okay, so guys, when we talk about this common bile duct, what is this common bile duct going to produce? It is going to produce your bile. Again, this is going to allow all the bile and pour it out over here into the duodenum. Okay, fine. But now, guys, what is this bile? Where is it coming from? How is it going to be? So, what did we say? Where is bile being produced? Bile is being produced in your liver. And how is this generally? Guys, your bile is going to be somewhat yellowish green in color. It is yellowish green. Guys, actual bile is going to be like, dark greenish. When will you see this thing? Guys, if you have suffered from loose motions, okay, so what happens the moment you defecate, guys? You must have noticed that those loose motions basically they smell awful, they smell bad. Again, if your normal fecal matter, if you check that, guys, that is not that smelly, but your loose motions, it is a dark greenish or it is in darker color, and plus it is smelly. It is because it is going to have this bile. What happens under normal circumstances? The bile gets mixed with the undigested food, and then it is going to be worked upon, broken down by microorganisms, and then converted from the yellowish green to brownish color. But in loose motions, you don't have that much amount of time. You have had something and immediately, right? So there is no time to act on the bile, and that is why you will see it is going to be dark yellowish green or dark greenish. Okay. Now, okay, so it is going to be yellowish green, but what does this contain? So if we talk about its contents, once again, guys, this is going to have water. Okay. Second important component, it is going to have cholesterol. Guys, this is very, very important. Cholesterol. And along with this, we have bile salts and bile pigment. But bile salts, guys, what are these? There are two bile salts, sodium taurocholate and sodium glycocholate. Sodium taurocholate and sodium glycocholate. And when we talk about bile pigments, guys, we have two bile pigments. We have bilirubin and biliverdin. Okay, bilirubin and biliverdin. Okay, fine. But now, what is this, bile going to do, guys? What is the role of bile? So let's try to see what are the reactions because of this. The first thing, guys, see, what did we say, guys? See, we said at this moment, we had this chyme. Okay, and chyme was acidic. Where was this chyme, guys? In the stomach. Okay, so here, we were seeing that, oh, we were having acidic chyme out over here. Okay, so we were having acidic chyme. But now what happens in the presence of your bile? Now, this acidic chyme, this is now going to be converted into your alkaline chyme. So acidic chyme is going to be converted into alkaline chyme. Now, how is it? Guys, you are going to see that during this reaction, you are going to see huge amounts of sodium bicarbonate being added. So this is in the presence of bile and NaHCO3. Guys, this NaHCO3 sodium bicarbonate, I'd already told you, this is nothing but natural antacid of the body. Okay, so acidic chyme is converted into alkaline chyme. Now the question arises, why is it so? Why do I want alkaline chyme? So okay, either here in color nahi dikh raha hai. Okay, so I'm just showing you. See, from here you're going to see just below. Okay, out over here, you are going to see alkaline chyme. Okay, right in the duodenum itself. The reason for this is, guys, all the other enzymes which are going to come after this, like, whether it is the pancreatic juice, whether it is going to be your intestinal juice, all of them, they work only and only in the alkaline conditions. They don't work under acidic conditions, and that is why it is very important that we convert the acidic chyme into alkaline chyme. Okay, fine. Chalo. Next thing, guys. Next thing, what we are going to see is we are going to have fats. Okay. Now, if we have fats, okay, I mean, when we talk about these, guys, at this moment in your stomach, fats were like huge molecules like this. Okay, they were huge. But now, in the presence of your bile, okay, now let's try to see. So, in the presence of bile salts, especially, okay, so if we look at these bile salts, that is your sodium taurocholate and sodium glycocholate, guys, these bile salts reduce the surface tension of the fats. And because they are reducing the surface tension of fats, what are they going to do? They are going to convert these fats into emulsified fats. They convert it into emulsified fats. But how are these emulsified fats? Guys, these are going to be nothing but smaller droplets like this. These are emulsified fats. Guys, just to give you an idea, how does this thing really look like? See, guys, you must have applied oil to your hair sometime or the other, right? So when you apply oil to your hair, guys, after that, when you try to wash your hands, when water falls onto it, and if you're like scrubbing your hands, immediately you'll see those small bubbles, globules, droplets being formed, right? That is nothing but your emulsified kind of appearance. Okay, so those bubbles kind of thing. This is what we are actually looking at. Okay, everybody okay with this thing? Guys, this is what is going to happen with regards to your fats. Is everybody okay with this thing? Just have a look, see what has happened. See, after this acidic chyme, immediately this acidic chyme was converted into your alkaline chyme. Why is cholesterol present? Because it is a component of your bile, because your liver is going to handle huge amounts of cholesterol. That is why. So basically, when we look at cholesterol, na, like always remember, cholesterol is not a bad thing, because cholesterol is responsible for giving you different kinds of your hormones as well. So all your steroid hormones, like whether it is testosterone, progesterone, estrogen, okay, all of them, they are going to come out from your cholesterol only. Plus cholesterol is also going to be responsible in the formation of your precursor of vitamin D, and ultimately it is going to help in the absorption of your the calciums and ions onto the bones, making them really strong. So that is why cholesterol is present out over there. Okay, because it comes from the liver. Liver handles a lot of cholesterol. Okay, everybody all right? Yes. Okay. So now, guys, what is happening? See, what is our aim? Our aim is to bring about digestion. So slowly, we need to cut down the food properly. Okay. Until now, let me just show you once again. Okay. Until your stomach. Okay. Until your stomach, what were the two reactions? I mean, the two things that had happened. Your starch had converted into maltose, and the second thing, like we saw, was proteins were converted into proteoses and peptones. Okay, so until here, that is the thing. Now we need to break them down further. See, we have carbohydrate, fats, proteins. We need to break down everything into its smallest forms, okay, into its monomers. We need to do that. So now let's try to see the next thing. Okay, so now, guys, what happens? Now we are once again in the duodenum only, guys. Okay, we are in the duodenum. So let's try to see now what happens. So we are going to have the pancreatic juice. Okay, and let's try to see what happens with regards to the pancreatic juice. Okay, now guys, I'm going to tell you to remember it in some fashion. Okay, I want you to remember the pancreatic juice as ants. Okay, why? We will see this. Okay, so now guys, when we talk about pancreatic juice, where is the pancreatic juice going to be coming from? See, we said we have your pancreatic lobules or your acini. So from your acini, this is now going to enter. So in that, right in the center, guys, if you remember, so let me just go back and show you. Okay, right in the center, we had the space for your pancreatic juice. See. See, in your acini, right in the center, we have pancreatic juice, right? So that is going to, take all the pancreatic juice, and then you are going to see that this pancreatic juice is going to come up through the pancreatic duct. And through the pancreatic duct, it is now going to enter into your duodenum. Okay, so this is how it is going to be. But now, guys, I told you, how are you going to remember this pancreatic juice? You are going to remember this as ants. Okay, let's try to see why. Okay, so first thing is A, guys. A is what? A stands for amylopsin. Okay, amylopsin, or the other name for this is pancreatic amylase. Okay, amylopsin or pancreatic amylase. Now, guys, whether it is pancreatic amylase, whether it is salivary amylase, whether it is any other amylase, intestinal amylase, guys, amylase will work as amylase only, because see what had we said, these enzymes, they work in the same fashion, they work on the same product, and I mean the same compound, and they will always give you the same end product. So whether it is your pancreatic amylase or whether it is your salivary amylase, their action remains the same. Okay, so now what happens? See, guys, whatever starch that was not broken down in your mouth, now that is going to be broken down in your duodenum because of the pancreatic amylase. So what do we say? We are going to say leftover starch. Whatever starch that was present, so leftover starch in the presence of amylase, this is pancreatic amylase, or you can say this is going to be your amylose. Okay, either of the two things you can use. Okay, pancreatic amylase or amylopsin, what is this going to get converted into? Once again, it gets converted into maltose. The reaction remains the same, because whether it is amylase from anywhere, it has to give you the same end product all throughout. Okay, so this is our next thing, guys. Okay, one moment. Some गड़बड़ हो गया. Okay, so this is the next reaction that we need to see, guys. Okay, so amylopsin. But now, after your amylopsin, guys, we are going to move on to the next one. Now, what is the next one? That is going to be your trypsin. Okay, but now T. Okay, so this is trypsin, and then there is your nucleotides also. Nucleotides we'll see a little later. Okay, but now we are going to have your trypsin. Okay, sure. But now, what is this trypsin actually produced as? Trypsin is actually produced as trypsinogen. Okay, pepsinogen, trypsinogen. Guys, whenever you have something with gen means it is a generator. Generator meaning it is something inactive. Okay, so now let's try to understand what is happening out over here. So trypsinogen, this is going to be inactive. Okay, so what happens is, guys, we have this trypsinogen, which is inactive in the presence of enterokinase, an enzyme. Okay, inactive thing. This in the presence of enterokinase, or we also call this thing as enteropeptidase. Enterokinase or enteropeptidase, what is this converted into? This is now converted into your trypsin. And guys, this trypsin, this is going to be active. So trypsinogen into trypsin. Okay, but now guys, what is this trypsin going to work upon? See, trypsin is going to work upon proteins. But until now, what do we have? See, guys, in your stomach, we had said that, oh, the pepsin had worked on proteins to break it down into proteoses and peptones, right? But what do you think? Are all the proteins really going to be converted? No, right? So the reaction is not going to be so specific that everything is going to be broken down right at the same time. No. So guys, now what are we going to see? So we are going to have proteins plus the proteoses plus the peptones. Whatever that had happened in the earlier case, these in the presence of trypsin, these in the presence of trypsin now is going to be converted into smaller proteins. What are these smaller proteins called? We call them as polypeptides. We call them as polypeptides. So just to give you an idea of what has happened. See, earlier your proteins were this much. Okay. Then when your pepsin had acted onto it, now what happened? They were converted into proteoses and peptones. Now what is happening, guys? When trypsin has worked onto this, now you are seeing that, oh, they are broken down into these smaller parts like this. Okay, in the next case, these polypeptides will be converted into dipeptides later on. So in dipeptides, meaning you're just having two proteins, so it is going to be even smaller. And these dipeptides, they are further going to be broken down into your amino acids. And then we will get the complete breakdown of your proteins. Okay, everybody all right with this thing? Getting this part? Okay, now what happens? Okay, so guys, now we have got our polypeptides. But now, guys, here the situation is a little different. Now, what happens? The moment we have your polypeptides, now there is another enzyme which is going to be activated. Now, who is that? That you're going to see chymotrypsinogen. Okay, so let me just label these reactions 1, 2, 3. Okay, now when this thing is happening, guys, we are going to have chymotrypsinogen. Okay, chymotrypsinogen in the presence of trypsin. Okay, everything is happening in the presence of trypsin now. Chymotrypsinogen in the presence of trypsin, guys, this is converted into chymotrypsin. It is converted into chymotrypsin. Guys, once again, Chymotrypsinogen. See, it is ending with chain. So what did I tell you? This is going to be inactive. Chymotrypsin, this is going to be active. But now the moment we have chymotrypsin, what is chymotrypsin going to work upon? Very simple. See, we had got these polypeptides. So these polypeptides, they are going to be worked upon by chymotrypsin. So polypeptides in the presence of chymotrypsin, they are going to be broken down into dipeptides. I just now told you, two proteins. So this entire reaction is going to take place out over here. Chymotrypsin. Okay. So this is the second thing. Okay. But now I told you, so A, N, T, S, out of that A and T are done. Now we are going to move on to your N. Now guys, what is this N? N is for your nucleases. See guys, whenever you are having your food, okay, at that time you're also having a lot of nucleic acids. You're eating nucleic acids as well, because see, all the cells and all, they're going to be broken down, and the DNA and all is going to be extracted. Everything is going to come out, right? That DNA and all needs to be broken down as well. And for that, we require these nucleases. Okay. So let's try to see. Now, what is these? The N is for your nucleases. Nuclease meaning it is going to break down your nucleic acids. So first, let me just tell you what is this, how is this going to be, what really happens. Okay. So guys, okay. So let's imagine first thing. Okay, I'll just give you the reaction, then I'll explain you how it is going to work. So we have our nucleic acids. Okay, and nucleic acids are what? Nucleic acids are going to be DNA and RNA. These nucleic acids, in the presence of nucleases, they are converted into nucleotides. They're converted into nucleotides. Okay, so nucleic acids in the presence of nucleases converted into nucleotides. But what are these? Let's try to understand. So guys, when we talk about nucleic acids, let's say this is your DNA. Now it can be your RNA also. I'm not saying it is only your DNA. Both of them are nucleic acids, DNA and RNA. So see, your DNA is going to be something like this. Okay, right? Now let me just uncoil this thing. See, this is in the coiled format. So let me just uncoil this. So you will see that, oh, your DNA is going to be a double-stranded structure like this. And then you are going to be having these bonds in between at regular intervals like this, right? Now what happens, guys? You are going to see that, see, wherever these bonds are there, actually there are going to be these small repeating units of your DNA. And that is why only bonds are seen in those places. Now, what are these repeating units of your DNA called, guys? These are nothing but your nucleotides. These are nucleotides. So now what happens now? Like when these nucleases act on this thing, they are going to break down this DNA and they're going to release these nucleotides. And what is this each black box nucleotide going to look like, guys? You are going to see it consists of 3 components. So you have the phosphate group. Phosphate group is attached to pentose sugar like this, and pentose sugar is attached to nitrogen base. Pentose sugar is attached to nitrogen base. So see this each black box out over there, it consists of these 3 things: phosphate group, pentose sugar, and nitrogen base. This is your nucleotide. Everybody all right with this thing? Okay, now guys, okay, so this is, I'll just mention, so this is your nucleotides. Okay, but now I need to break these nucleotides properly, guys. Okay, I need to convert it into the smallest form. Okay. Okay, so this is your nucleotides. Okay, but now guys, we don't stop out over here. You are going to see some connecting reactions. So we have these nucleotides, guys. The moment we get nucleotides, nucleotides are going to be worked upon by nucleotidases. Okay, so nucleotides, they are going to be acted upon by nucleotidases and they are converted into nucleosides. Okay, we'll see what is it. Okay, so in the presence of nucleotidases. Okay, so see once again, nucleotides. See, I told you how is this nucleotide? It is phosphate group, plus it is going to be your pentose sugar, and plus it is going to be your nitrogen base. This is your nucleotide, right? Now, guys, what happens is when nucleoside, your tidases comes, these nucleotidases, they are going to cleave off only the phosphate group. Okay, so as the phosphate group is removed, now what are you going to see, guys? In your nucleosides, you have only two things remaining. So you are going to have one, your pentose sugar, that is your deoxyribose sugar in the case of DNA, and second thing, you are going to have nitrogen base. These two. This is your nucleoside. Okay, but now we are not going to stop over here. We need to break this down completely. So in the next case, guys, what are we going to have? You are going to have nucleosidases. So you have nucleoside. This in the presence of nucleosidases, this is converted into your sugar and nitrogen base. Okay, so you are going to completely break your DNA down. DNA or your nucleic acids. Okay, so once again, let's just have an idea. So if you talk about nucleosides, see guys, nucleosides, we said it is going to be like this. Now, when nucleosidases are going to jump in, guys, what are they going to do? They are going to cleave this bond, the glycosidic bond between the sugar and the nitrogen base. And once that is done, guys, both of them, they are free. So you have sugar and next thing you are going to have nitrogen base as well. So like this, sugar and nitrogen base. This is how it is going to work, guys. Everybody okay with this thing? Guys, just have a look at this. How is the sequence going to happen? So in your nucleases, first you are going to have nucleases, then you're going to have your nucleotidases, then you're going to have nucleosidases. Just have a look, guys, if everybody is okay. Nucleotide is the smallest repeating unit of DNA or your RNA. It is the smallest repeating unit. So it keeps on repeating every now and then. Okay, everybody all right, guys? Sure, can we move ahead? Okay. Do these reactions occur at specific pH? Yes, I told you here it is going to be alkaline pH. So if you're talking about this alkaline pH, then it is going to be ranging from 8 to around 11, 8 to 12 in that range, guys. Okay, see, whatever reactions that were taking place in your stomach, they were in the acidic pH. Whatever reactions that are taking place in the duodenum, it is in alkaline pH. And why is that? Because your acidic chain was converted into alkaline chain with the help of sodium bicarbonate. And that is why it is that way. Okay, chalo. Now guys, let's try to see the next one. Okay, the next one is your S. So see, out of ants, now we are done with A, we are done with N, we are done with T. Now the last is your S. So what is this S going to be? So guys, the S is actually your stipsin. Okay, stipsin, or we also call this thing as pancreatic lipase. Okay, pancreatic lipase. Now, guys, lipase, lipids, fats. So this is the one which is going to act on your fats. So, okay, but now, until now, what fats were we carrying, guys? See, in your bile, what fats were converted? Emulsified fats. So now these emulsifieds are going to be broken down by this pancreatic lipase. So let's try to see what is the reaction. So you are going to have this emulsified fats. In the presence of pancreatic lipase, this is going to be converted into its smaller components, that is fatty acid plus glycerol. You cannot break down fats any more than this. Okay, so fatty acids plus glycerol, and this is happening in the presence of pancreatic lipase. Okay, or STIPs. This way, guys. This is what we need to know with regards to your entire pancreatic juice. Once again, guys, I'm asking you to remember this as ants. Okay, just remember ants, amylopsin, nucleases, trypsin, and stipsin. Now, guys, remember in trypsin, you always need to have the chymotrypsin also. Okay, because both of them they go hand in hand. That is why. But then just remember this as ants. Okay, fine. Chalo. So now, okay, yes, no issues. Okay, now let's try to move ahead, guys, and let's try to see the next part. Okay, so now, guys, where are we? We were in the duodenum. Okay, so we were in the duodenum. Everything was happening in the duodenum. Now, from the duodenum, guys, we are going to go down into your ileum, in your ileum region, guys. Okay, so let's try to see. Now, where are we? See, now we are going to have this entire duodenum, then we have your jejunum, and then we have your ileum. So let's imagine we are in this region, guys. Okay, so let's say this region. So this is our ileum. Okay, now in ileum, guys, what are we going to see? We are going to see that the ileum produces its intestinal juice. Okay, so the next thing is intestinal juice. Okay, now what happens, guys? Let's try to see this thing. So before we talk about what is this intestinal juice and what is really going to happen, how is the breakdown going to occur, let me just give you a little bit idea. See, guys, when we talk about carbohydrates, okay, carbohydrates, these are generally divided into your three main types. Okay, that is monosaccharides, we have disaccharides, and we have polysaccharides. Okay, mono, di, and polysaccharides. Guys, monosaccharides, what are we going to see? We are going to see monosaccharides, we have glucose. Second, we have fructose. And third, we have galactose. Okay, these are the three monosaccharides. But now, guys, when we talk about disaccharide consists of two units. Okay, so if glucose plus glucose takes place, guys, so if you have glucose plus glucose, this is going to result into maltose. If glucose binds with fructose, glucose plus fructose, then we get sucrose. And if glucose binds with galactose, okay, so glucose plus galactose, Then we are going to have your lactose, that is your milk sugar, that way. Okay, so guys, this is how your these things are going to be. Okay, and when we talk about polysaccharides, okay, in polysaccharides, guys, we are going to have cellulose, we are going to have glycogen, and we have starch. Okay, these some examples you always need to remember, guys. Okay, now see, our idea is what, guys, in your body. Okay, when we talk about your intestines, you are going to find these maltose, sucrose, lactose, and your aim is to break them down into their individual components: glucose plus fructose, glucose plus galactose, that way. Okay, so this is what is going to happen in your ileum also. Guys, how are you going to remember this intestinal juice? You need to remember this as melli. Okay, guys, once again, melli, konitari melli doesn't make sense, but it will help you to remember. Okay, so now what is this? What is this melli? What is really going to happen? Okay, so first thing, guys, this first M, that is going to be for the enzyme maltase. Okay, maltase. Guys, as the name suggests, maltase, what is it going to work upon? It is going to work upon your maltose or malt sugar, right? So immediately you are going to say, we have maltose. Maltose in the presence of this enzyme maltase, okay, in the presence of maltase, now, what is going to happen? This is going to be broken down into its smallest units. And what were its smallest units? See, guys, what is it made up of? Glucose plus glucose. Immediately, glucose is made available to the body. Now, either you can store this glucose or you can use it for respiration. That is depending upon your body. That way. So, this is how glucose is going to, this is how maltase is going to be. Okay, fine. Second one is E. E is for your erepsin. Okay, E is for erepsin, or this is also going to be for dipeptidases. Okay, dipeptidases meaning it is going to work on your proteins. See, until now, see. Come here. See, we had got dipeptides because of the activity of chymotrypsin, right? Now, time it is to break it down into its smallest unit. So, erepsin, guys, this is going to break down dipeptides. Dipeptides in the presence of erepsin, this is converted into your amino acids. So guys, see, you are actually started to break down everything into smallest unit. This is also smallest unit. This is also smallest unit because you cannot break down amino acids anymore further. Okay, this is in the presence of dipeptidases or erepsin, whatever you like to call it. Okay, fine. This is second. Okay, then third thing we are going to have L. Now, L is for what, guys? L is for lipase. Oh, but now we had already seen lipase. See, once again, out over here, guys, we were already seeing lipase, pancreatic lipase, Stifson. So now let's try to see, guys. So this lipase, whatever we're talking about, lipase will work on lipase, meaning it will work on lipids only. Lipids, which lipids we are talking about? We were talking about your emulsified fats, right? So here also the same thing, guys. Either it is emulsified fats or whatever leftover lipids. Lipids in the presence of lipase, this is now going to be converted into fatty acids and glycerol. Once again, see guys, this is going to be the monomer because I told you we cannot break them down even further after this. Okay, chalo. So this is also the smallest unit. The next L is for your lactase. Okay, but once again, lactase or lactases. Okay, now once again, guys, this is going to work on what? It is going to work on lactose. So lactose in the presence of lactase enzyme, What is this going to break the lactose down into? Very simple. This is going to break it down into glucose plus galactose. Glucose plus galactose. That's it. Okay. Next thing. Okay. The last one, guys, we have I. I is for your invertase. Okay. Or we also call invertase as sucrase. Okay, and what is this going to do? See, guys, if it is sucrase or invertase, sucrase is going to work on sugar. Sugar is nothing but your sucrose table sugar. So you are going to see that we have this sucrose. Sucrose in the presence of sucrase. Okay, sucrase, or we can say invertase, whatever you like to call it. Again, this is going to be ultimately broken down into what, guys? See, this is going to be broken down into glucose plus fructose. Glucose plus fructose. That's it. That is what is going to happen. And see, guys, if you look out over here, we have got smallest units, the basic units of all the things out over here. Whatever the breakdown was taking place, see, glucose, amino acids, fatty acids, glycerol, glucose, galactose, glucose, fructose. You cannot break this thing down any more further. And that is why, guys, what do we say? That after this thing happens in your ileum, our digestion is complete. And you know what, guys? After digestion, okay, after digestion, we are going to say, see, before digestion, we said we had your chyme, right? The acidic chyme converting into your alkaline chyme, but it was chyme at the end of the day. So after digestion, guys, we had this chyme. But now after digestion, chyme is converted into chyle. Okay, so always remember when we talk about chyme, chyme is in the stomach and the initial parts of the intestine. Whereas when we talk about chyle, chyle is going to be in your intestine, especially in your ileum region. Okay with this thing? Fine. So guys, this is all what we need to know with regards to the different reactions. Okay, different reactions present in your uh, digestive system. So guys, just have a look at this. Just see if everybody has understood what has happened. Just have a look. Okay, everybody all right, guys? Sure, no issues. Understood this part properly, guys, how your digestion has taken place? Guys, at least answer yes or no. I'm not able to see anything, guys. Hello? 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 Mic check. Okay, 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 chalo. But now, guys, after this, let's try to see. Okay, let's try to see some of the hormones of the gastrointestinal tract, guys. Okay, let's try to figure out what really happens out over there. Okay, so guys, see, as I told you, like, these enzymes, these hormones, okay, whatever, basically, see, enzymes are not going to work on their own. Okay, they require certain hormones to help them being produced and released at certain specific times. So, what are those? Those are nothing but the hormones of the gastrointestinal tract. Okay, so let's just try to see what are the hormones of the GIT. Okay, GIT is gastrointestinal tract. Okay, GIT. Okay, so now, guys, there are actually four different hormones which are going to play an important role. Who is producing this thing? This is going to be done by your stomach and intestinal mucosa. Okay, so now, first thing, guys, who is the first one? The first one, we are going to see it in your stomach. And what is this? This is gastrin. Okay, so gastrin is going to be produced in the stomach, okay, by the stomach walls. But now, the moment we have gastrin, guys, what is this gastrin going to do? This gastrin is going to stimulate the secretion of HCl. Okay, so it helps in the secretion of HCl, and plus we are going to see pepsinogen. Both these things, they are going to be secreted by your gastrin. Okay, but after your gastrin, we have secretin. Okay, so what is the secretin going to do, guys? Secretin is going to act on the pancreas. Okay, so acts on exocrine part of the pancreas. Okay, and now when it acts on the exocrine part of the pancreas, guys, what is it going to do? It helps in the secretion of NaHCO3, sodium bicarbonate. First, okay, it helps in the production of sodium bicarbonate, and at the same time, it helps in the formation of pancreatic juice, release of pancreatic juice. So two things, guys. Okay, helps in the secretion of NaHCO3 and helps in the secretion of pancreatic juice. Okay, third one. Third one, guys, we have cholecystokinin. Okay, cholecystokinin or CCK. Okay, cholecystokinin or CCK. Now, what is this going to do, guys? This cholecystokinin, this is going to act on two things. One, it is going to act on the gallbladder. And now, see, cholecystokinin. So, cholesterol, it is going to come from the liver, or it is going to be coming from your gallbladder. And chole is what basically chole means your bile. Okay, bile contains cholesterol. I had already told you this thing. So, you are going to see that this gallbladder, this is now going to help in the secretion of bile. Okay, so cholecystokinin, we have gallbladder. Gallbladder helps in the secretion of bile. And now the second thing that is out over here, it is also going to act on the pancreas. And when it acts on the pancreas, it asks the pancreas to produce the pancreatic enzymes. That way. So that is cholecystokinin. Okay. Finally, guys, we have the last one out over here. That is GIP, gastric inhibitory peptide. Okay, gastric inhibitory peptide, GIP, or the other name for this is enterogastron. Okay, or enterogastron. Okay, but now what is this? See, gastric inhibitory peptide, meaning it is going to inhibit something. So what is this going to inhibit? So if we look at this, guys, it stops the gastric secretion, meaning first, the one that was happening in the stomach, it is going to stop that. It stops gastric secretion. Okay. And second thing now, guys, you're also going to see that this prevents motility. Okay, motility or mobility of food in the intestines. Now, why? Because see, if the food is continuously moving, will will you be really able to absorb the nutrients? No, because they are like moving away. Whatever little a bit of amounts of nutrients you can absorb, that is fine. But maximum you won't be able to absorb. And for this, we have this DIP. It prevents the motility of food in the intestine so that you can absorb or so that you can have increased absorption. So has to have increased absorption. Okay, that's it. That is what we need to know with regards to the hormones of the GIT, gastrointestinal tract. Just have a look, guys. Just have a look. Okay, chalo. So now, guys, let's try to move ahead and let's try to see the next thing, guys. Okay, so guys, we are completely done with your small intestine, but still the large intestine is remaining. We haven't really spoken of the large intestine. So let's try to see what exactly is going to happen in your large intestine. Okay, sure. So once again, let me just quickly just draw this thing. Okay, so let's imagine we had your stomach, something like this. Okay, and now let's imagine. Okay, now I'm just drawing it as single thing. Okay, let's say this is your small intestine, and now your small intestine is going to be converted into your large intestine. Okay, so something of this sort, guys. Okay, so now what happens? See, when we talked about the stomach, okay, in your stomach, guys, what was present? It was your chyme. Let me write it this way. So you had chyme out over here. Okay. Now what happens is, guys, from the stomach, when we have this chyme, from this chyme, what are you really going to reabsorb? You are going to absorb huge amounts of water, plus you're also reabsorbing huge amounts of glucose, and along with that, you are reabsorbing inorganic salts. Now, guys, always remember outward arrow meaning you are absorbing, inward arrow meaning you are putting it something. Okay, that is how we generally do. Now, guys, all this thing is going to move through your small intestines and comes into your large intestine. But what is the large intestine going to do? Guys, what did I tell you? Is the large intestine really going to help you in digestion? No, this is not going to help in digestion. But what is it going to do? It is going to convert your chyle. that is the semi-liquid or semi-solid kind of mass, whatever that is there, it is going to convert it into your solid fecal matter. But how is that possible? That is possible because here in your large intestine, you are going to have maximum absorption of water. Okay, so you are like continuously removing water, like wherever the food is entering, like undigested food, you are continuously removing water. And as you're continuously removing water, by the way, sorry, where are we going to remove the water from? From the child. Okay, so child was in your intestine. So from there, it is going to enter into your large intestine. You're removing the water quick, 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 quick. Okay, and now you are going to see that by the time it reaches out over here, you are going to have completely undigested food. Okay, this is your undigested food. Okay, and now what is going to happen when this rectum is going to be full? Once again, the body is going to send impulses to the anal sphincter. Okay, and anal sphincter is going to open up under the influence of the spinal cord. Once the job is done, it is once again going to be restored back. So once again, it is going to shut down. That is nothing but your anal sphincter, and the entire process is called as defecation. Okay, so whatever that has happened right now, this is known as defecation. Okay, so either you call this thing, or the second term we've already seen this, we've already spoken it or as egestion. That's it. That is what is going to happen with regards to your large intestine, guys. Okay, and if you want, I'll just add two more things. From the small intestine, you are going to see maximum absorption of food taking place. Maximum absorption of food. And guys, in the same region, what have we also said? You are going to see the lacteals of the small intestine. And lacteals, they have only one job. What was their job? To absorb fats. Okay, so this is how it is going to be. Guys, just have a look. So with this, we are done with the entire digestion process. Okay, everybody all right? Fine, let's move ahead. Yes, 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 yes or no? Quickly, quickly, quickly. Okay. Okay, now guys, the next question arises is what is going to happen to all these things that are being absorbed by the body? Okay, so whether it is your carbohydrates, whether it is your fats, whether it is your proteins, okay, what is going to really happen to those? Okay, so what happens is, guys, they are going to undergo a process of assimilation. And assimilation means whatever you have absorbed, okay, into the blood, ultimate aim is to help it get converted into protoplasm inside the cell. That's it. That is assimilation. Okay. So we have assimilation. Now, what is this, guys? This is nothing but conversion. Okay, conversion of absorbed substances into protoplasm of the cell. Again, that is nothing but assimilation. So what will happen to the glucose that is absorbed? That is either going to be used for your respiration, or it is going to be given to the cells. The cells may, they may help to store it in the form of glycogen. So this is once again nothing but your assimilation. You're going to use these raw products, and ultimately you're going to create. Just a moment, just a moment. Okay. Chalo. So guys, that is assimilation. Okay, but now guys, the next thing that we are going to talk about is a little different aspect. We are seeing that okay, assimilation, the food is going to be absorbed, and it's going to be taken up by the cell. It's going to be converted into protoplasm. Some of it is also going to be used for generating energy. For example, glucose, it helps to generate energy. So now we are going to see the next thing is what happens to these compounds? What happens to these products? How are they going to provide energy? So guys, the next thing we're going to talk about is what are calorific values? Okay, so basically, guys, you must have always heard that oh, food contains these many calories, this contains that calories, chocolate and fatty foods, high calorie food. So we are trying to find out how is this calorie actually determined. Okay, so let's try to figure this thing. So guys, we have calorific values. Okay, so when we talk about these calorific values, guys, there are actually two types of calorific values. One, we have gross calorific value. Okay, gross calorific value. And the second one, guys, we have physiological values. Okay, gross calorific and physiological values. Now, guys, when we talk about gross calorific values, what are gross calorific values? See, always remember this gross calorific value is the one which is going to be the amount of heat liberated under ideal conditions, meaning in your proper experimental conditions, not in your day-to-day life. So guys, you must have heard that your cars have some mileage, right? So for example, you are going to have, let's say, maybe like a 15 kilometers per liter or 20 kilometers per liter. But see, sometimes on your television sets and all, they show you ads, and in their ads they claim, oh, you are going to get 27 kilometers per liter. Hello, can you hear me? Hello, mic check. Hello guys, can you hear me? Hello. Yeah. Zui, can you hear me properly? Or else just log out and log in again. Others can hear me properly. Okay. Okay. So guys, see, when we talk about these, mileage and all, 27 kilometers per liter or 30 kilometers per liter. But in real life, in reality, are you going to really get that much? Guys, how much is the mileage that you get, in your day-to-day life? It might be just 15 kilometers or 14 kilometers. This is also, I'm talking about like, very good mileage. Okay. Just to give you an idea. So when they say 27 kilometers, it is under experimental conditions, under controlled conditions. Same way, when we talk about the gross calorific value, this is uh, under entire experimental condition in control conditions only. Okay, so let's try to see, guys, what is this? If I have to define this, this is nothing but the amount of heat liberated by complete combustion, by complete combustion of 1 gram of substance, 1 gram of substance in a bomb calorimeter. Okay, now what is this bomb calorimeter? Bomb calorimeter is an basically it is, a setup. Okay, so basically it is going to be like a setup like this, and now you are going to keep your food whatever that is there, and then you're going to burn this thing, and then you try to find out how much amount of calories are going to be generated. This is your bomb calorimeter. Okay, but now see, when we talk about calories, okay, how is calories determined? We always say kilocalories per gram. This is the general notion. Okay, but now let's come to physiological value. See, guys, I told you, in your ads, they are going to claim some else mileage, and in the reality, you're going to get something else. So, what are these physiological values? Physiological values is the same thing, amount of heat liberated, but under normal conditions. Okay, so let's try to see what do I mean by this. So, this is nothing but the actual energy or the heat liberated. Okay, actual energy which is liberated by combustion of 1 gram of substance, okay, under normal conditions, okay, not in your bomb calorimeter. Okay, so once again, guys, how are we going to keep this thing in mind? How much energy? It is kilocalories per gram. So let's try to see, guys, what are the different components and how much they are going to produce. So, guys, first one, we are going to have fats. So if you look at fats, guys, the gross calorific value, this is going to be really, really high. 9.45 kilocalories per gram, a really, really richest source of energy. And now, if we look at physiological values, that is under your normal conditions, it is just going to be 9 kilocalories. Okay, fine. Second thing, let's say we are having your proteins. Okay, so when we talk about proteins, guys, proteins are going to have a gross calorific value of 5.65. 5.65. And under normal circumstances, this is just going to be 4. Okay. Third, we are going to have your carbohydrates. So carbs. Now, carbs, they are going to give you 4.1 kilocalories as gross calorific value. And once again, under normal circumstances, it is just going to give you 4 kilocalories only. This is how it is going to be like, guys. Okay, everybody all right? Just have a look, see if you're okay with this. Then we'll talk about something that is absorption of fats. Okay, with one very small part. Just have a look. And guys, in your exams, whether it is your NEET exams or whether it is your normal college exams, they will play with these values, okay? And they will try to confuse you with the values. You need to be completely aware of this. Okay, and then generally they will always ask you, what is the gross calorific value? Because the other one, the physiological values, you are quite aware of it since you are, fifth or sixth. But then these gross calorific values are a little different. So keep this thing in mind properly. Okay, chalo. So let's move ahead, guys, and let's try to see this small part. Okay, absorption of fats. How does this really happen? So, guys, when we talk about fats, so we had said your fats they are going to be like this. Okay, then this is going to be converted into your emulsified fats. Okay, so these are emulsified fats. They are going to be even smaller droplets like this. Okay. Now what happens is, guys, this emulsified fats, these are actually going to be further broken down before absorption into smaller pieces. Those smaller pieces, we call them as micelles. Okay. But, okay, just a moment. Just a moment. Ah. So now these are converted into these micelles. Okay. But now still I cannot really absorb this thing. So guys, now what happens is these micelles, they are going to be coated, okay, with your proteins. Okay. So let's see, this is going to be coated by some proteins like this. The moment it is coated by proteins, we call them as chylomicrons. These are termed as chylomicrons, guys. Okay, so chylomicrons. And now these chylomicrons are going to be absorbed by the lacteals of your small intestine. So let's say this is the villi, and now inside we are going to have the lacteals. So this is then going to enter like this. This is how that entire process is going to work. Okay, everybody all right with this? Understood this part properly, guys? Okay. So guys, once again, what are we going to do is for this time, okay, we are going to stop out over here. Next class, okay, guys, I want you to go through all these reactions, okay, and you need to know them by heart. So once again, first thing that we're going to do in the next class is we'll once again walk through these reactions, and then we have certain diseases associated with your the food and calories and all. So for example, we have marasmus, we have kwashiorkor, indigestion, constipation, all those things that are there. So that we'll talk about in the next class. Okay. Chalo. So guys, we'll stop over here for the day and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye.